Hi everyone, my name is Danny. Today I'll be talking about protein synthesis. I'll discuss how it works, what it means, and why we need protein synthesis. First of all, synthesis means to make something. So protein synthesis basically means to make protein. And I'll also talk about how protein is made. There are many different eye colors in the human race, such as green, blue, and brown. And the eye color is determined by DNA from parents to their children. A parent's genetic makeup determines the pigment amount, or melanin, in the iris of his or her child's eye. For example, with high levels of brown melanin, the eyes will turn out to be brown. If there are high levels of green melanin, the eyes will turn out to be green. If there are high levels of blue melanin, the eyes will turn out to be blue. Now let's talk about DNA versus RNA. DNA means deoxyribonucleic acid, while RNA means ribonucleic acid for short. DNA has instructions an organism needs to develop, live, and reproduce. These instructions are found inside every cell that is passed from parents to offspring. Deoxyribose is double-stranded and has a double helix structure, as you can see in my photo right here. The double helix right here. And it is the sugar with the bases adenine, thymine, cytosine, and guanine. You can see them like adenine and thymine pairing with each other, cytosine pairing with guanine, and these are called base pairs. While RNA is similar to DNA, it has some subtle differences. The RNA's job is to produce proteins, carrying the messenger instruction from DNA, which itself contains genetic information that's needed for the development and maintenance of life. The ribose is the sugar where the bases are adenine, uracil, cytosine, and guanine. And the A pairs with the U and the C pairs with the G. RNA are single strands that contains single nucleotides attached to the structure, as you can see right here. Now let's talk about mRNA. mRNA stands for messenger RNA. So in order for a DNA to leave the nucleus, the mRNA was the DNA in this situation. mRNA is a single-stranded RNA molecule that is a letter that contains genetic information that will lead to the nucleus. They go to find the ribosome for it to produce proteins later on. As you can see, it's basically a letter here, like I drew, that contains information. The tRNA means transfer RNA. tRNA is the type of RNA molecule that helps to code a messenger RNA sequence into a protein. It is a member of a nucleic acid family called ribonucleic acids. This tRNA molecule has a purpose. It is to read the mRNA strand to match its anticodon to a special codon of mRNA. This would start to build a protein. This is the rRNA, and it is short for ribosomal RNA. It is in the ribosome, and its function is to direct the translation of mRNA into proteins. For everyone that is wondering, hey, why is protein synthesis so important? It is so important because it helps prepare and build your body's tissues. It allows metabolic reactions to take place and coordinate your bodily functions. This would also provide your body with a structural framework to maintain proper pH and fluid balance. Now let's talk about transcription and translation. These are the two main steps of protein synthesis. If you don't know which step goes first, there's a really easy way to remember it. There is a C in transcription and there's an L in translation. A C comes before L in the alphabet. That's why transcription is the first step of protein synthesis and translation is the second step. It's just an easy way to remember it. And transcription is when the RNA polymerase goes into the DNA strand by unzipping it and attaches to it. And it slowly reads the DNA strand, becoming mRNA, messenger RNA that has important information about the DNA. Next, the mRNA matches the base pairs like adenine and thymine together. But instead of matching adenine and thymine, it goes with uracil. It always goes in a 3' to 5' direction, and it can't go out the other way. And finally, the mRNA leaves the nucleus through nuclear pores and into the cytoplasm to look for a ribosome. Then comes translation. mRNA then attaches to the ribosome to be read and finds a start code on 3 bases, AUG. The first amino acid, AUG, is brought in by the tRNA, and anticodons on it will match the codons on the mRNA, releasing an amino acid every time it does, which creates a polypeptide chain that are proteins. This will continue it until it reaches stop codon UAC and will release into the cytoplasm as a result. Thank you for watching my video, and I hope you enjoyed what you watched, and see you next time.